Dissolution test results evaluation for alcohol dose dumping. Dissolution testing is required to be performed with alcohol in the dissolution media in different percentage for certain formulations. Mainly extended release, sustained release or prolonged release type of formulations are required to undergo the dissolution testing and dissolution comparison with alcohol in the media. Welcome dear viewers to this video. This will be a short video to understand how the dissolution results are evaluated or studied or compared to demonstrate the resistance to alcohol dose dumping. The extended release formulations and some delayed release formulations or modified release formulations are required to undergo this testing. Also in this video we are going to learn about the dissolution comparison, dissolution testing requirements, criteria provided by FDA and the EMA. Let's start with the basics. So dose dumping means sudden release of the API or drug or more than the specification release. The modified release formulations are designed for site specific drug delivery and also for the sustained drug delivery. But if there is a dose dumping or sudden release or more release of the API, that means the release is hampered, design is not as per the requirement, and also due to the rapid release it may show toxicity and the side effects. So to determine whether the controlled release formulation behaves as required for safety and efficacy when the alcohol is consumed or not, that is studied in the dissolution testing for alcohol induced dose dumping. It is abbreviated as ADD, alcohol induced dose dumping. Also, the study is required to perform or to prevent rapid release of or potentially harmful drug release dose dumping that could lead to overdosing. Key parameters for evaluation are the rate of drug release, then comparison of drug release and the threshold for dose dumping. Rate of drug release measures the amount of drug released over the period of time in alcohol containing solutions. Comparison involves comparing the drug release in alcohol media versus standard dissolution conditions without alcohol. Threshold for dose dumping, that means significant deviation from the expected release profile indicates potential dose dumping. For example, I can say that, or you can consider that if you are having a specification of not more than 25% in 1 hour, and if the formulation shows 50% or 60% drug release in alcoholic media, then that means there is a rapid or more release of the drug and it is showing the dose dumping. So this dose dumping is required to be studied and there should not be dose dumping in the alcoholic media. Interpretation of the dissolution results. So first is normal release profile and versus it will be a dose dumping. So normal release profile, that means slow and steady drug release in the alcohol media. No significant difference from the standard dissolution conditions. That means the product is safe and there is no dose dumping. Then second is the dose dumping. If the product fails for dissolution, that means there is a dose dumping. Rapid or accelerated release of the drug in alcohol media. Release exceeds expected levels or it release occurs too quickly. So this type of formulations are unsafe for consumption with alcohol, and also these are not meeting the quality requirements with the reference formulation. So these can be considered as unsafe. And unsafe formulations indicates potential for overdose or adverse effects. Then coming to the quantitative evaluation, cumulative drug release over time. So in this we can plot the cumulative amount of drug released versus time for the dissolution testing containing media with and without alcohol, so that we can clearly understand what is the difference. Criteria for dose dumping. So now coming to the criteria, when you can say that there is a dose dumping and when you can say there is no dose dumping. So a significant spike in the release profile or curve when alcohol is introduced, exceeding a predefined threshold for drug release. Example, more than 50% release in a short period, like within 1 hour. Also, you can say that if the predefined specification is not meeting, if the formulation is showing higher percentage of drug release than the specified range, that means there is a dose dumping. Example of evaluation, standard release profile versus alcohol induced dose dumping. So for standard release profile, gradual increase in the drug concentration in the dissolution medium is the standard release profile, while rapid rise in the drug concentration, possibly exceeding safe therapeutic levels, is the alcohol induced dose dumping. Impact onto the formulation. What will be the effect or impact of dose dumping onto the formulation? So formulation will be at risk if the formulation fails, and if the formulation passes, then there will not be significant risk. Formulation at risk. If dose dumping is observed, the formulation may need reformulation to prevent alcohol related safety concerns. Formulation that passes. Formulation that show no significant difference or that still have controlled release in alcohol may be deemed as safe for consumption with alcohol. But generally, alcohol should be avoided with medications. Whenever we are prescribing or using or we are dispensing the medications, always there should be a concern for the alcohol consumption with the medicines. Alcohol consumption should be avoided with the medications. Regulatory guideline for evaluation. FDA regulations are there. Alcohol dose dumping testing is required for specific drug formulations like modified release formulations, controlled release formulations, and some of the delayed release formulations. Results are required to meet the strict criteria to ensure patient safety. And labeling section should be there, which provides you clear indication for positive dose dumping findings that might lead to additional warnings or contraindications related to the alcohol consumption while you are taking the medicines. And now coming to the very important part of this video, US requirements versus EMA requirements. So I have studied different guidelines and I have find out some differences. So you can refer this slide or this information whenever you are asked to provide information on the differences between US and EMA requirements. So as per US, dissolution media is mainly 0.1 normal HCl with 0% ethanol, 5%, 10%, 20% and 40% ethanol. While for EMA, it may be a QC media or it may be a 0.1 normal HCl dissolution media with ethanol concentration of 0%, 5%, 10% and 20%. But in US, 40% is included. In US, QC parameters can be evaluated for dissolution media and as per the product specific guideline by the FDA. If your QC media is not 0.1 normal HCl, then other medias you can provide the information. Time points 2 hours and sampling time point every 15 minutes for US, while as per EMA, the early time points for dose dumping testing should be done and QC time point should be provided. US requirements are comparison by F2 value, comparison with the reference formulation, and comparison with the dissolution profile without alcohol. Then, as per EMA, it will be a comparison by F2 value and also specification of QC testing will be performed. As per US and EMA, both requires data of the mean, minimum, maximum values, percent RSD on 12 units data or 12 units testing. For example, tablets or capsules with 12 units. Reference profile for USFD is dissolution without alcohol in the media versus dissolution in media with a different percent of the alcohol. And alcohol means it is a ethanolic solution or ethanol. As per EMA, reference profile will be the dissolution profile of reference without alcohol in media versus dissolution in media with different percentage for the test product. So these are some of the differences and similarities. So I hope this video will help you to understand about the alcohol induced dose dumping, dissolution testing for alcohol induced dose dumping, and comparison between the US and EU requirement. Thank you for watching the video, and let's meet soon with such kind of informative video.